Dr. Ross Chambers, who is the Vice President of Antibody Discovery at Integral Molecular, with over 20 years of antibody discovery experience in both academia and industry. He focuses on discovery of antibodies against multi-pass membrane proteins, where he directed the discovery of commercial antibodies, thousands of commercial antibodies using DNA immunization, B-cell cloning, and other isolation techniques. And before that, he developed DNA immunization for large-scale antibody isolation projects at UT Southwestern. He earned his PhD from University of Otago, New Zealand, and completed his postdocs at UC Davis in Berkeley.
We're an antibody discovery company, and there's lots of antibody discovery companies out there. We are different in that our specialty is we focus on making antibodies to a very difficult class of proteins called multi-pass membrane proteins. These are the proteins that weave through the membrane multiple times. They are proteins such as GPCRs and ion channels, and they've been very difficult to make antibodies to. Just a little about our company. We're a Philadelphia-based company. This is us. Oops. This is us here, ninth floor. We've got space over here. And our company focuses on membrane proteins and primarily on antibody discovery and characterizing antibodies to membrane proteins. We've been around for almost 20 years, and even though we're in industry, we still publish a lot. We do a lot of high-profile publications, mainly on our virology work. A lot of our antibody therapeutic work we can't publish. Just a quick overview of what we do. So we are an antibody discovery company. We have an antibody discovery technology we call MPS that goes all the way through from discovering targets all the way through to producing antibody therapeutics ready for preclinical studies. And we have a number of other technologies inside there. I'll tell you about live particles. This is a way for epitope mapping and a way for testing specificity that I'll go into. Okay, so first about the problem. So multi-pass membrane proteins are a very important class of drug targets. They are the leading class of drug targets, but most of the way that these are addressed are with small molecules. And the problem with these proteins, though, is that a lot of these drugs that are developed, small molecule drugs to these targets, a lot of them fail in clinical trials. This is from a study a few years ago where 62% of small molecule drugs fail in the clinic because they hit other targets. They have off-target safety issues. Another 20% fail, they bind the right target, but they bind the target in the wrong tissue. So most drugs fail in the clinic because of safety issues. Now, antibodies could solve this problem. Antibodies are known for being much more specific, but the problem with this problem is that it's very difficult, or it's been very difficult to make antibodies to these kinds of proteins. So there's about 2,400 multi-pass membrane proteins in the human genome, but only 3 of them have been targeted with antibody therapeutics. But many of these have been targeted with small molecule drugs. Now the reason these are so difficult is this is a picture here of a typical multi-pass membrane protein. This is a lipid membrane. Here it weaves through the membrane multiple times. The reason they're very difficult to make antibodies to is that there's very little on the surface for an antibody to bind to. Often many of them have very small extracellular regions. They're very complex proteins. The extracellular part is made up of discontinuous segments from this protein weaving through, and they have lots of post-translational modifications decorating them. So they're very, very complex. A key thing is the protein structure, the native protein structure, is dependent on the lipid membrane. So this means that you have to maintain this protein in the lipid membrane through all the steps of manipulation. They're very difficult to make antibodies to. They're very generally poorly immunogenic, and this is because they're very difficult to express at high levels, or when you do express them, they're toxic and kill the cell. and many of them are very highly conserved, and that reflects their very important physiological role in the human body. And so because they're highly conserved, that means that when you inject them into an animal, they're seen as self and they don't react well and make antibodies. So that's the problem. This slide really summarizes my journey in this. I've been working in this area for 20 years, and I've focused on making antibody drugs to multipass membrane proteins. And this is I've tried everything, and this is really the synthesis of what we have tried and what doesn't work for making these antibodies. So a very common approach to making antibodies is you synthesize a peptide, or you'll make a recombinant protein in E. coli and purify it. Those approaches don't generally work, and the reason is they don't have native conformation. As I said, these proteins require the lipid membrane for structure, and these don't have that structure. So, I forgot to mention another immunogen is cell immunization, but that isn't as effective because there's very small amounts of the antigen on cells, and there's many other proteins on those cells that are immunodominant, so you don't tend to get a focused response on the target. So what we have focused on is DNA immunization, that I'll go into, and lipid particle immunization. It's a combination that's been very effective. Another issue is where you get your antibodies from. Very popular, it was really that industry is split between using animals or using naive synthetic libraries, using phage display. However, we've focused on animals. Really, this hasn't proved effective for multipass membrane proteins because these antibodies are generally relatively low affinity, and the amount of protein we have available for panning the libraries on is very limited, and it's usually inefficient to enrich those antibodies out. So generally, this has been ineffective for pulling out antibodies for multipass membrane proteins. So we've focused on animal-based methods for discovering antibodies. And then there's the MAB discovery method itself, how you pull the antibodies out. Hybridoma, obviously a very popular method, but really it generally produces a very limited number of antibodies. So we don't really use that. The workhorse we've been using is phage display. That pulls out large numbers. And in development, we're working on B-cell cloning. And really, the industry is moving towards B-cell cloning. This is a big move in the industry. So this is our platform we use for discovery. We take a target. Now, you skipped over the step very casually, as many people do when you make an antibody. So they say, oh, we just get the antigen and we stick it in the animal and make an antibody. Well, that very first step is a showstopper for membrane proteins. You've got to really think about your antigen. So we use, as I said, DNA and live particle immunization. We work very hard to make this antigen. It's got to be native, and it's got to be in high concentrations and immunogenic. We also, like many people, we can immunize mice and rabbits. They're popular models for making antibodies in animals. But most of our work has been in chickens, and I'll just tell you why that's required for being successful here. Once we've got immune responses, we move into phage display, and we make immune libraries. We pan on life particles and pull out the antibodies. We use a lot of automation. And oftentimes we're looking for agonist or antagonist antibodies, which are very rare. They usually only make up a few % of the antibodies that you generate. And then, as Yiquan mentioned, we have to engineer these antibodies often, and we have to humanize them, affinity mature them sometimes. And if we want to make bispecifics, we have a common light chain that enables us to do that. So anyway, a lot of people say they can make antibodies to multipass membrane proteins, but really we're different in that we have a very robust system. We hardly ever fail. It has a very high success rate at making large panels of antibodies. Okay, so just a few words about the conventional approaches. So if you're making an antibody normally, you would get some pure protein, first maybe made in E. coli. You would stick it with some adjuvant like froins. You stick it in a mouse, and you would make antibodies. But as I said before, this doesn't really work for membrane proteins. The reason is, as I said, a lot of the epitopes of this protein are non-native, so they're irrelevant for a therapeutic. This will also focus a lot of the responses on internal epitopes, which are again irrelevant for a therapeutic. A lot of these membrane proteins have large intracellular regions that aren't therapeutically relevant. And a lot of the epitopes are cryptic in the sense that they're either highly conserved, which means that you'll make antibodies to the target, but you won't make them to the spot that you want, the functionally relevant sites. Or they're in little pockets on the protein, and you want an antibody to reach into that pocket to have an effect. So our approach that's been proven to be successful, we use chickens, and I'll say why. For the immunization, we use DNA and life particles for the immunization, and we have these special adjuvants that not only produce high titer responses, but they're non-denaturing. They don't denature the antigen. And this is what enables us to get most of the antibodies that come out of our programs are hitting the relevant epitopes, and it's a matter of then sorting through these for the therapeutic ones. Okay, so why do we use chickens? So really it's because a lot of these proteins are highly conserved and you won't get an antibody response. Most mammals are pretty closely related. So mice and humans, are 92 million years separated, and a lot of the proteins between those are pretty similar. You can jump into other animals, but really you're not changing the story much. Rabbits are about as different as mice are from humans and as from llamas. In any mammal, they're all pretty much the same distance. But if you go to chickens, there's a really large evolutionary distance, and the proteins are very different from humans. And that means that they'll see these human proteins as foreign and respond to them. And you get much more diverse immune responses. And chickens, even though they're very divergent from mammals, they still make antibodies. And these antibodies are pretty much, the antibodies itself are conserved in mammals. So there's essentially the same molecule that a human makes, so we can put them back into humans. But at the same time solving this problem. This just shows you quantitatively. This is drug targets in particular. This is the top 600 drug targets that people are chasing. And you can see here that over half of half of drug targets are more than 90% identical to mouse. So that means that for half of drug targets, you're going to really struggle to make a good, diverse antibody response. Same for rabbits. But in chickens, almost all the proteins are not highly conserved, and you can make good immune responses. A few words about one of our technologies, lipoparticles. This is actually what founded our company. This was an invention that our founder, Ben Durant's, developed at University of Pennsylvania, and this is what we used to spin the company out 20 years ago. So lipoparticles are basically virus-like particles that contain high concentrations of the membrane protein. So we express the membrane protein on a cell, a mammalian cell. We express the viral machinery that buds off from the cell and makes these particles that have very high concentrations of the membrane protein. The protein is in its native conformation, so we make relevant epitopes. And because it's a virus-like particle, the immune system loves these things, and they're very immunogenic. When we immunize chickens, we actually, as I said before, we use a combination of immunogens. And we start with DNA immunization. I'm not sure if everyone's familiar with DNA immunization. I came from the lab that invented this. Actually, it was at UT Southwestern, Stephen Johnson, who developed this technique where you, instead of delivering a protein into the animal, which most people do, you deliver an expression plasmid that expresses the gene. So this plasmid gets transfected into the animal's cells. The animal expresses the protein on the surface. It's in the membrane. It's in its native conformation. So it's really a killer application for immunizing with multipass membrane proteins. And it's highly pure. It's the only thing you're delivering is just this. Now, DNA immunization is a very powerful method for getting immune responses, but oftentimes it drives it into memory B cells and not antibody secreting cells, and that's what you need to detect titer. So oftentimes you immunize with DNA and you won't see an immune response in the animal. Even though it's made memory cells, you can't see it. And what we find is you have to pop it with a little bit of protein, and we use light particles for this. And if you prime the animals with DNA and then boost them with light particles, you get this great immune response that works for multipass membrane proteins. Here's some examples. So we've done many, many different programs, but here's some examples of a number of different classes. So a claudin, an ion channel, a GPCR, and a transporter. All of these thread through the membrane multiple times. All of these are highly conserved. You see, look, it's 95, 96, 97, 95% identical between human and mouse, which means that a mouse is going to have a tough time making an antibody to this. But in chickens, it's much reduced. But when we immunize all these proteins into chickens, you see all of them give these great immune responses that are reacting to native epitopes. This is flow cytometry, so we're seeing native epitopes being generated and antibodies recognizing that. The flip side of proteins being highly conserved amongst mammals is that we can often get an antibody that is pan-reactive across multiple mammalian species. And in drug development, you want to, your preclinical studies, you need to test your antibody in various animal models, and it's very nice if your antibody that reacts with human also reacts with mouse, so you can use that same antibody in the preclinical as the clinical studies. Otherwise, people have to make another antibody, a surrogate antibody, that reacts with the mouse one to do those studies, which doubles your amount of antibody discovery work. Fortunately, because mammalian proteins are highly conserved, chickens, it means that chickens can make antibodies that recognize across all these species. These are very popular, preclinical animal models, and as I showed you before, most mammalian proteins are highly conserved, so you can get antibodies that cross-react. And here's an example here. So here's a number of proteins that are highly conserved between human and rodents, and we've immunized with the human version. We've made antibodies to the human version, and when we test those antibodies on the rodent, you see that they also react well with rodents. And now we have an antibody that we can use for clinical studies and for preclinical, avoiding the need for a surrogate. And then one final plug for, sorry, I don't mean to speak about chickens so much, but we do use them, and we're unusual in that we use chickens, but they really are wonderful sources for antibodies. But one of the, chickens make very normal looking antibodies relative to mammals, but one unusual thing about chicken antibodies is that the business end of an antibody, the heavy chain CDR3, the part that really drives most of the interaction of an antibody, that's the most variable in length. And you can see in this little graph here in blue, this is the length of CDR3s from all the therapeutic antibodies out there. Most of these came from mouse, although it's starting to shift to human more. People are doing more human antibodies, which tend to be longer. But most mouse antibodies are relatively short, around 10 amino acids. What that means is that the topology of an antibody from these things is flat, generally flat. So if antibodies with this length here are generally flat. Now that's fine. Flat antibodies are great. They bind lots of things. But it's a problem when you want to access epitopes that are in pockets, because there you need something protruding to reach in to bind that pocket. And you can see here on this graph here, this is data we generated using NGS of B cells, looking at all the chicken, millions of chicken sequences. You can see here that chickens have much longer CDR3s. So many of the antibodies in chickens have these longer CDR3s. They have a protruding CDR3 that can reach into pockets. These have been, the literature has followed this a lot, mainly led through the llamas. Llamas also have produced these long CDR3s, and they've been very useful.
If you look at the sites on antibodies where anti-drug antibodies pop up, does that cluster in one particular region? Like, does that worry you? I guess.
That's a good question. Yeah, I don't know the answer to that actually. Yeah, but we can discuss it at the end. As I was saying, llama antibodies make long CDR3s, and they have been shown to bind into pockets in this crystal structure. This is a GPCR, it binds in a pocket here, and this is an antagonist. It locks into a certain state. And over the years, we've done a lot of programs developing antagonists and agonists, and you can see a lot of these have long CDRs, although we haven't got crystal structures to show how they bind. But we suspect that some of these are binding in pockets. All right, so then let's move to panning and screening. So when we get immune responses in chickens, we create a phage display library. So we isolate the B cells, we PCR out the genes, and display them as SCFEs on phage display. We pan them on light particles. You can pan these on anything like cells and things, but we found that a critical step is having high concentrations of the membrane protein to really efficiently enrich the antibodies out. And we use light particles, which has been very effective at enriching them. And then once you've panned them a couple of rounds, you'll then express the soluble scFvs and test them we use ELISAs on light particles to find all the antibodies that are specific to the target and then validate on flow cytometry. We use a lot of automation in our workflows for deep screening. We do up to 10,000 or more immunoassays, 3 to 4 well plate. We use a lot of, I love this machine here, the ViaFlow. It's been a game changer for us for high throughput screening and robots for picking clones and reformatting. Should also mention humanization. So because these are therapeutics that are going to go into a person, they touches on the question you just asked, is they can't be immunogenic. So we have to humanize them. It turns out that this can sometimes be difficult, but it turns out for chickens, actually, it's relatively easy. Chickens only have a single antibody framework, like humans, so mice, for example, have like 50 or more frameworks. So everyone's a different problem. Chickens only have one, and the framework that's most closely related to human is this very popular VH323, which is in many therapeutics, and less so the VL325. But these are already in clinical antibodies. The frameworks are highly conserved to human, so it's not a big jump to humanize them. And this is some examples. We simply do a CDR graft onto the human framework, and most of the time we have very minimal loss in affinity. That's usually the problem with humanization, is you graft CDRs across and you lose affinity. But here you can see the parental chicken antibody and the humanized one, and you see very minimal loss in affinity, and that's typical.
These data are humanization just by CDR grafting?
We also put in some back mutations occasionally. Yeah, but most of the time it's a simple graft with a sprinkling of back mutations. And this works for about 75% of the cases. Doesn't work for everything. There's a few that don't work, but most of the time they humanize just fine. All right, so I want to talk a little about characterization of antibodies. So specificity is really important. Remember that the whole point of us doing this work is we want to make better drugs than small molecules. Small molecules are really struggling with specificity. Antibodies are better at specificity. They have more surface area, obviously, to engage the targets. They can be more specific. But you don't get a pass. I mean, antibodies can have cross-reactivity problems. We actually have developed a technology to test this called the membrane proteome array. What this is essentially all human membrane proteins, about 6,000 genes, that we expressed in cells, in mammalian cells. We format them in 3D4 well plates, and then we can screen an antibody across all of those 6,000 proteins and measure them by flow cytometry to determine whether it binds the target. Of course, it should bind the target, but does it bind to anything else? You'll see here an example here. So this is the 6,000 membrane proteins here, all the data points here. And this is the binding, and you see it should bind the target, but what we're looking for is, are there any off-target binding events, which would cause a safety issue. As I showed you before, this is a major problem with small molecules, off-target binding. But we've been using this. We launched this as a service a couple of years ago for other people to use, and we've tested hundreds of antibodies from various therapeutic programs. And the big surprise for us, and this is supported by other people in the field, is that a quarter of the antibodies that we test have off-target binding. So antibodies, yes, are better at specificity, but you really got to test them. That they really have major concerns. So you've got to test them for specificity. Here's an example from one collaborator we had. They had 2 lead candidate antibodies to this target. We ran them on the membrane proteome array. This one on the left here, it bound the target and nothing else. But this one here, in addition to the target, it also hit this other protein. This other protein isn't a paralog. It's got no sequence identity to this target. It's completely different. Now, the interesting thing is we told them this, and then they injected these antibodies into mice to see, where do they bind. This target that they were going for was supposed to be expressed in the thyroid. So they saw for this antibody, it went to the thyroid as you expect. This one, when they injected into the mice, they saw it went to the thyroid, but they also saw that it went to the adrenal medulla. And this target in the annotation is supposed to be expressed in the adrenal medulla. So it was a nice confirmation of our platform.
Is your readout dependent upon cell-killing capabilities built into that C region, or
to the what?
Is your readout for off-target binding dependent on cell death and the ability of the antibody to induce cell death, or?
No, it's flow cytometry, so we're just measuring binding.
Oh, so, okay.
Just binding, yeah. Okay, so that's really our technology platform. Last part of my talk, I'm just going to talk about some examples that we've used our platform to discover therapeutic candidates. We work with partners. We work with a lot of pharma companies to develop antibodies for them. We also have our own internal pipeline. This is our internal pipeline here, and I'll go through a couple of examples. We'll see how much time I have. Okay, the first target I want to talk about is CB1. This is a GPCR. It's the cannabis receptor. This is the one that makes you high. It's a validated target for a very important disease called NASH that probably everyone's been hearing about. NASH is this new epidemic in the country that is linked to obesity. What happens is, if you are overweight, you accumulate a lot of fat in your liver, and this fat in the liver can drive this inflammatory response that eventually makes the liver fibrotic and damages it, and you eventually end with cirrhosis and you need a liver transplant. Now, this is a huge opportunity out there. There's no therapies, and you'll see just about every day a new clinical trial reporting out where a NASH drug has failed. People are very actively pursuing NASH as an opportunity. There was this fantastic small molecule drug, an antagonist called rimonabant, that was developed in the 90s and approved. And it had wonderful effects. People lost weight. It affected metabolic syndrome. It was a really incredible drug. But of course, CB1 is expressed in the liver where it has those wonderful therapeutic effects. But it's also in the brain where it makes you high. But this is not an agonist. This is an antagonist. So this made people sad and suicidal. And this is what caused the withdrawal of this drug from the market. So what we're hoping to do is to make an antibody antagonist to this target, because an antibody can't pass the blood-brain barrier, so it won't hit the brain CB1, it will just hit the liver CB1. Okay, so this is a profile of this target as for making antibodies. It's very difficult. So it's a GPCR, has 7 TM domains, so it's very structurally complex, as I mentioned. It's toxic when overexpressed. It has a very large N-terminus, which is therapeutically irrelevant. The epitopes there aren't therapeutically relevant, so this is going to be immunodominant. And then it's 97% identical to mouse, so it's very difficult to make responses in mice. To make an antagonist, you could make an antibody that simply blocks the ligand binding. But in this case, that's an unlikely mechanism because the ligand this is a top-down view of the crystal structure. And it's been crystallized in the antagonist state in blue or the agonist state in orange. And what they've shown is the ligand, the hypothesis is that the ligand, the natural ligand, binds through the plasma membrane. It's a very hydrophobic ligand. And so the ligand's coming in through the side and binding in the site. So an antibody is not likely to block the ligand binding. What we really have to do is lock it into the antagonist state. We want to lock it into the blue state and prevent it from switching into the orange state. But that's a rare antibody. Okay, so here's a summary of the campaign. As I said, we had to engineer the antigen to make it express well. We immunized chickens, and we got titers in most of the chickens. We put it into phage display, a couple of rounds of panning, screened some clones, got hundreds of hits, and sequenced those and boiled down to 108 unique families that we focused our search on. We express these as IgG1s. We need a fairly large amount of antibody in order to do functional studies. And then we produced them in CHO cells and did functional screening to find antagonists. Here's the functional studies. We ran all of the 100 or so antibodies through functional assays. In this case, it was a calcium flux flipper assay looking for antagonism. And we had 4 antagonists come through, and this one here, IM102, is our lead candidate. This antibody is pretty good affinity, 2 nanomolar, which is pretty decent for an antibody. Most of the time for chickens, we get picomolar to nanomolar affinity. And of course, we ran it on the MPA to make sure it's specific, and it's completely specific. It has no other off-targets, including other cannabinoid receptors, CB2 or GPR55. And then we need to show that it does something. And the animal models are problematic, actually. It's difficult. So the one thing we thought we would do is look to see if we could do primary human liver cells to test this efficacy. So the key cell in the liver that's driving a lot of this disease is the cell called a stellate cell. This stellate cell expresses CB1, and when there's a lot of fat in the liver, it triggers this inflammatory response, and it becomes activated, and it causes inflammation and fibrosis. And that's the thing that is supposed to be driving this disease. So what we're hoping to do is add an antibody to this to shut this cell down that should either revert back to its inactivated state or undergo apoptosis. So what we did is you can buy primary human hepatic stellate cells. We activated them in culture and then treated them with our antibody drug, and then monitored fibrotic markers or inflammatory markers using qPCR. And you show the result here. This is an isotope control. And then treatment with IM102 reduced collagen and actin and an inflammatory marker, TGF-beta1. All right, so that's one story. Let me switch to another story real quick. This is a cancer target, Claudin-6. This is an interesting protein that it is not expressed in normal tissue, but it is expressed in a number of cancers: ovarian, lung cancer, endometrial cancers, and stomach cancers. This target is also very difficult, multipass membrane protein. It's highly conserved to mouse, 95% identical to mouse. And then the kicker here was it has a very close paralog called Claudin-9. And Claudin-9 is almost identical to Claudin-6 on the outside, except for 3 amino acid residues. So we need an antibody that hits 6 but doesn't bind 9. Here's that challenge here. So Claudin-6 is a member of a family of Claudins. Claudin-6 here, it's got this close cousin 9, and also not far away is Claudin-3 and 4. And here's those 3 amino acid differences I told you about. Actually, this one here is really not very accessible. We've got to distinguish this guy up here, Q156, and maybe R145. There's also an allelic variant in the human population. Some people have isoleucine here, and some people have valine, so we want it to be neutral across that. We don't want to read out on that. So that was the challenge for us. So we did an immunization campaign. We didn't have to actually engineer this antigen. It expressed quite well. We immunized animals, got good responses. We did panning, and here we did panning differently in that we had light particles with Claudin-9, and we used those to deselect. We used them to scrub out all the antibodies that would bind to 9. We screened a few thousand clones, got hundreds of hits, and that boiled down to 68 unique families. We expressed these in CHO cells, and then we got 4 highly specific ones. I'll show you in the next slide. So here is the binding and flow cytometry on transient transfected cells expressing 6 or 9. So in gray is the reactivity to Claudin-6, and all of them bind Claudin-6 as you expected. But in red is the ratio of 6 over 9. So we want a high ratio on 6, very minimal 9. So you can see here that a lot of the antibodies leaned towards 6, but still had very significant 9 binding. But we found a very small number that had very selective binding. That presumably the antibody was homing in on that amino acid difference to discriminate those molecules. Here's it showing a titration against the the close related claudins, so 6, that's the target, this is the binding here, but 9, 4, and 3, it's not touching. Now there is a clinical benchmark. Someone, Ganymed, a German biotech company that was then bought by Astellas, has developed a therapeutic antibody as well. But they developed theirs in mice, and we compared our molecule to that benchmark molecule. And while they both had similar claudin 6 binding, theirs still had significant claudin 9 bindings. That's a potential safety risk when they put that into clinical trials. Again, we tested for specificity, and our antibodies are very clean on claudin 6 and not seeing claudin 9 or any other molecule in the human proteome array. Okay, so the last story I want to talk about, this is one that has been a 10-year journey for me. I've been working on this target for 10 years trying to make antibodies to it and failed. I've thrown the kitchen sink at it, and we finally had a breakthrough just last year. So KV1.3 is an ion channel, and it's a very interesting target for autoimmune disease. So when a T cell, a naive T cell, activates and does its job, it upregulates KV1.3, and it's required to maintain that cell in its activated state. And of course, effector memory T cells are driving a lot of autoimmune diseases. So the idea is to shut this target, this ion channel, down. So we don't want to kill the cell necessarily, although some people are looking at that approach, but what we want to do is shut it down. There is some drugs that have been developed out there. These are peptide toxins from various weird creatures, I think, I forget where that was from. But these small peptides have shown therapeutic effect. These are peptide inhibitors of this, but have stumbled in the clinic because of half-life and bioavailability issues. So that's really been quite difficult to overcome. And last I've heard, I think they've abandoned this as a therapeutic entity. So we're thinking that an antibody inhibitor to this target will have obviously have great half-life and bioavailability, and should solve this problem. Okay, so this protein is one of the most difficult targets I've ever worked on. It's a 6-spanning membrane protein. It's a tetramer. 4 of these come together to form the structure. It's highly conserved, of course, to mice. It's very, very low expression. It's the most difficult protein to express. It's miserable. And the target we really want to hit is the voltage gate. This is a voltage-gated ion channel. So when the membrane potential changes, this thing opens up. And it senses it through this domain here, which has a bunch of arginines in it, and it senses the charge change and it shifts up. So we're looking for a conformational blocker. It binds one of the conformational states. Here's a summary of the discovery campaign. We had to engineer this antigen to get it to behave. We immunized chickens. Actually, I've immunized mice and rabbits with this thing. Nothing at all happens. No immune response at all. Only in chickens have we seen an immune response. We made libraries, panned them, usual screen, pulled a bunch of hits out, and expressed the antibodies and found 3 inhibitory leads. Here's some of the characterization of these molecules. This is just simply binding and flow cytometry on transient transfected cells. We've also tested them on Jurkat T cells. They naturally express KV1.3, and you can see these antibodies are binding. And also we bought some human primary T effector memory cells and stained the antibodies of those, and we can see it binding to primary human cells. And then we've tested, this is the functional part. Can it inhibit? This assay is not patch clamp, but this rather unusual dye binding assay. And here is a positive control, which is Shaker toxin, which is related to that delazotide thing that inhibits. And this is one of the antibodies showing dose response inhibition of this channel. And here's the titrations for a number of our lead antibodies. That's the last example. Yes, so anyway, that's our story. We're antibody company that just focuses on these difficult targets. And what we're hoping to do is opening up these multi-pass membrane proteins for therapeutics so that we don't have to just rely on small molecules, but expand it into antibodies, which really have a lot of exciting capabilities. And that's our company, our team. Blue is our theme color, so we all dress in blue and orange for the company photo. Yeah.